Good afternoon, ladies and gentlemen. It's a great pleasure to be here. I'm really delighted to know that so many basic scientists and researchers are here to talk about the stem cell, which is a pretty new area for some people, and there are like some experts like Dr. Pidon is there. So I'm actually, my graduation is in microbiology. My PhD is in stem cell biology. So from 2005 and 2015, I was working with the stem cells, with the culturing of these cells. And for the last 6 years, I'm working on the clinical side, assisting the clinicians in different applications of stem cells. So today, I'm going to talk about the MSCs, SVF, and Nanofat, their differences and application. There was our discussion on Nanofat, so I will try to clear some of the things related to that. Basically, my postdoc is in tissue engineering that I have done from Japan. And in that, I'm working on the skin grafts, and I have developed the products from the Mesenchymal. And they are in clinical application in 2 centers, one center in Peshawar and 1 center in Islamabad. So in next talk, maybe in like near future, I may be talking about the application of tissue engineering and also the stem cells. So the field of stem cell is exploding. Everybody is now talking about the stem cells because there are enormous applications. If you talk from the hair, as we have learned today, to the toe. In every field, in every aspect, like we talk about the myocardial infarctions, there are so many brain disorders, and everybody is talking about the stem cell. And the number of stem cell centers are growing and growing in each of the hospitals. Like in View Hospital, you have the center. In private sector, like University of Lahore established the center. And recently, all of the universities have established a big regenerative medicine center. So it's not only in the world, but also in Pakistan, this field is exploding. So if you talk about, many people have talked about the application of PRP. This is the PRP from 8 different ways from the same patient, and you can see the difference in all these preparations. Like in this, it's like yellowish, it's red, and mildly yellowish, and so on. So you can clearly see the difference if you use different, for example, centrifuges. Like is it a fixed or is it a free swing centrifuge? So you will get different results. Similarly, there are different, like it's very much depends on your hands-on practices. Like if you are going to prepare SVF, who is going to prepare? Who is going to use that? It very much depends on your handling. So how you are going to handle these kind of stuff. So these are the results and these are the published results. So you can see the variation. This is a common technique. Every clinician now, like even in the villages, they are preparing the PRP and using that. So if there is a variation in PRP, stem cells, it's a bit advanced than that. So there will be variation, and we will expect a variation as well, because these are not the pills, these are the biological things. Another thing, like this is the era of regenerative medicines. You are the clinicians, so in the future, you will be the cell biologists. You will be treating your patients with the cell biology and cell biology based products. So you have to focus on this area very much. MSCs, we have talked a lot. So many, like it's very hard to talk at the end because you are bombarded with a lot of information so far. So I will briefly touch about some of the things and the applications. If we talk from the bone marrow, we isolate the stem cells from the bone marrow. This was like a pretty famous area in the sense that initially, there was bone marrow transplantation was done for the cancer patient. So many researchers are familiar with this kind of technology. So they have isolated the mesenchymal stem cells from the bone marrow. We can also isolate from the adipose. Cord blood, as Dr. Pidon, he was talking about the cord blood. This is the future of stem cell therapy. So many centers across the world, they are making the products, and there are so many clinical trials going on across the world on the cord blood. So this is the future of stem cell therapy, because you can use, for example, the conditions like for the stroke or COPD and many other areas. Cord tissue is another source of mesenchymal stem cell. We can also isolate. The beauty of cord blood and cord tissue is that the stem cell we isolate, these are the mere stem cells. So if you put into the patient with some pathologies, they don't react. They don't actually, the grass versus post disease. So they don't have that kind of problem. And we can also isolate the mesenchymal stem cell from the adult tissues, like we can isolate from the liver, from the kidneys, we can isolate from, there are now cardiac stem cells, so we can isolate those stem cells from the cardiac tissue. I'm really delighted to be here today because in 2009, when I was working in the United States, I came back to Pakistan, I was working on cardiac stem cells. I presented in the very same hall on cardiac stem cell like differentiation process, and I won the best young investigator award back in 2009. So it's a been long journey working with these kind of cells. So if we talk about the bone marrow, for example, if we have the bone marrow aspirated, there are a number of things we can do. We have the aspirate, we can culture that, and we can have the hematopoietic stem cells and mesenchymal stem cells. Another famous thing that we mostly do, we have we use microfat. We do the centrifugation, we can isolate the mononuclear cells, that can be used as a therapy. Another thing that can be done, we have the bone marrow aspirate, and we can do the magnetic based cell separation. So these are the future technologies our scientists and our clinicians must be aware of. If we talk about the adipose-derived stem cells, people were happy with the bone marrow stem cells. Why they have shifted towards the adipose-derived stem cells? There are some things, there was a study published in Nature, reference there, because in bone marrow, there are mostly the progenitor of our blood cells. In our body we have 23 trillion red blood cells, and in every second 15 billion red blood cells are going to die. So there is a lot of replenishment of those cells. So the hematopoietic stem cells are mainly active in our bone marrow. If we talk about the adipose, the mesenchymal stem cells, they are more rich source and it's very easy to isolate. There were a lot of discussion, and Dr. Samhi was explaining about the liposuction procedure. It's a common practice for the clinicians now. So they can have the iliac cells, and these cells are clinically more active. So that's why the clinician researchers they were more focusing on adipose-derived stem cells. So there was some confusion in previous discussion. So if we have like the adipose tissues, so you will see different types of cells. The big cells, these are the fat cells, they are residing there. These cells are the progenitor cells. On the capillaries, these are the pericytes. The pericytes also have very similar biology as the MSCs, because if we study their markers, if you are a scientist and study the markers and the gene expression patterns of the pericytes and mesenchymal stem cells, they are pretty similar. So these are also there, and we also have the mesenchymal stem cells. They are also residing there. So I will talk when I will discuss more when I will be talking about the Nanofat. So there was discussion that we can isolate, we can prepare the stromal vascular correction. There are a number of ways. One is the mechanical way, another is the enzymatic way. People have discussed that we can use the collagenase in that, and there is also now the automatic options, and I will discuss briefly about this. There is also evolution of mechanical stromal cell isolation technique. Initially, what we used to do, we have the fat, we used to have two syringes, and we do the emulsification. There is just connector, and we used to do the emulsification of that. So that was back in 2013. Then lipogems is a very fascinating event. There are tons of studies on lipogems. So in lipogems, they have beads. So they put the beads into the tube, and then they shake it vigorously to break the fat into very small fragments. So that was the evolution that was done in 2016, and there is a lot of literature available on the application of these beads to break down the fat. And then with the evolution, we have the connector and we have the filters. So this being used recently, that we can connect the tubes, and afterwards we can do the filtration. So if we have to isolate the microfat or the nanofat, we can do the emulsification and we can do the filtration, and this is how we can do. The recent ones are the sharp layers. These are called the ionizers, and I'll discuss a bit more about these ionizers and how fascinating these are. So there is like enzymatic way. If we talk about the enzymatic way, we can do it manually. It will reduce the cost. I have done myself like over 100 manual preparation, and you also learning in your practical session that we can have the fat, we can digest with the enzyme, and then we can spin it to isolate the SVF as many people have shown. If you are not going to use this, there is another option. We can also have the, for example, these are the Stampia kits. In the Stampia kits, the procedure is the very same. Only this is the sterile system if you are going to use. So what we can do, we can put the fat, we can put the enzyme, we do the put into the shaking incubator so we can digest the fat, and afterwards we put in another tube, we do the centrifugation, washing and centrifugation, and we can have the SVF that is being shown there. So this is like, it's very much depends on you. If you are going to do the procedure in 50,000, you can do it manually. If you are going to charge over 100,000, then you can use the kits. So because kits are very expensive, we have to import. We are not like producing in Pakistan. So this is the method. Another, like I talked about the anodizer. This is the future. This is a recent technology that we get. There are different like anodizers of different sizes, like 4,000 micrometer, 2,400, 1,200, 600, 400, 200, and 100. So these are the very short blades that you can see over here. So what we have to do, these are also the connector base. We have one syringe of fat, we can have the agonizer attached to it, and we have another syringe, and we pass through these agonizers so we can break down the fat. So this is how we can prepare the, and the beauty of this is that if you have to go towards the nanofat, for example, or if you have to prepare the SVF, you can do both using these kind of agonizers, and this is what we are also using in our center. And there is also option of, like if Professor Fridon, he has talked about that you can have a setup of in like 5 million rupees, and if you are going to spend like 50 million or more, you can have a simple machine. And this machine has the beauty that it can do everything for you. You just have to put the fat inside, and at the end you can have the SVF with your, so you can put in your operation theater, and afterwards you can get your SVF. But the problem with these kind of setup is these machines are pretty expensive. The tubing and all the like usable, these are very expensive. So in Pakistan, I think our patient might not afford it. There is another option. This is a tube prop that can also be used in your operation theater. On one side, you are going to isolate the fat, just like you do the liposuction. You put inside on the top chamber, you control the temperature with this machine, and it also mixes for you, and then it goes into the lower chamber. It goes finally into the last chamber, and there is a filtration process, and at the end you can get the SVF in just 1 hour. So this is the thing, something you can put in your operation theater. You just have to put the fat inside, and then you can get the SVF. So this is very simple. Like you don't have to set up the laboratory. Just have this machine, and it will do everything for you. So the things are like becoming very easy for the clinician. So you should be very happy with that. There is a lot of definitions for the nanofat and microfat. So you can see there are over 50 different types of like fat. You like talk about the bean size fat, macrofat, microfat, nanofat, millifat, micronized fat, supercharged fat. So there is a lot of definitions, and it's actually there are different procedures. I will show in the next table in which they have like this type of different procedure. Either it's enzymatic procedure or it's a mechanical procedure, and the way to produce, like if you are going to mix the fat with EPP, that's one way. That could be like prepare a supercharged fat for you. Are you going to prepare like your SVF or the fat, and you are going to mix the ERP with that? So what type of fat we are going to produce? So QT fat and there's lots. So there's a lot of confusion in that as well, because there is so many things.